Hi again, guys. So, I'm going to do a quick rundown of the non-Mendelian inheritance patterns for those of you who are still a little bit foggy on what they might be. I think most of you got a pretty good grasp of them, but can't hurt to have a little review. And so, these are inheritance patterns that do not fit the traditional Mendelian setup of having one trait with one gene, having two alleles, one of those alleles being dominant and one of those alleles being recessive. Any variation in that setup constitutes a non-Mendelian inheritance patterns. And so I've arranged them in what I think is the least complex to most complex order. And so codominance or codominant inheritance, right? That's pretty straightforward. I still have two alleles, okay, but they do not have a normal dominance recessive relationship. Okay. In a heterozygous organism, both alleles will phenotypically be present. Okay. And so if my dominant allele is red and my other dominant allele is blue, okay, my organism would be red and blue. I'd see red, I'd see blue. Okay. Incomplete dominance is a very similar situation, okay? But instead of seeing them together, they blend. Okay? So in a heterozygous organism, if first allele is blue, second allele is red, my heterozygous organism is going to be purple. It's a blend. Multiple allele is simply any trait that has more than two alleles available. So I might have a purple allele, a red allele, and a yellow allele. Okay. And so I set up little Punnett squares just to show how this can impact the way that inheritance occurs, right? So like I have full different setup. And so I can have a baby that is a genotype that's entirely different than either parent. That's possible with multiple allele traits. Okay? I mean, that's also possible with regular allele traits too. So anyways, sex-linked. Okay. This is really straightforward, not overcomplicated. Okay. People tend to overcomplicate this one. This one is just the gene that we're looking at. It's on the X or the Y chromosome. That's what makes it sex-linked. If it's on the X chromosome, we call it X-linked. If it's on the Y chromosome, we call it Y-linked. Well-named things. Okay. Now, because females have two X's and males have XY, this impacts the way that any particular gene is inherited based on whether or not the offspring is male or the offspring is female. Okay. So for X-linked traits, the males are hemizygous, meaning they got one copy of the gene that we're looking at, and the other chromosome is a Y. It doesn't have that gene. Okay. Different shaped chromosomes, different genes. The female just has a normal genotype. She's either homozygous or heterozygous. Nothing's different there. Okay. And then the offspring, these ones would be female, and these ones would be male. And so in this case, this particular Punnett square that I set up, the inheritance isn't any different because everything's dominant. Right? But it can impact the way that recessive traits show up. Recessive traits will show up more readily in a male for X-linked than they will for a female because the male doesn't have that chance to cancel out that recessive trait the way the female does. For Y-linked, the males are again hemizygous. They got one copy. Okay? But females have 0 copies. They don't have any genotype for this, right? Because they don't have any copies of this gene. Okay? So Y-linked inheritance is really easy. The males are always going to be exactly the same as whatever their father was, and the females aren't going to have the trait. That's it. Easy peasy. Linked genes are when the chromosomes are physically, not the chromosomes, the genes are physically on the same chromosome. Right? So when we do meiosis and independently assort our homologous chromosomes, that doesn't matter for linked genes. These genes are always going to move together. Right? So our Punnett square predictions do not work for linked genes. That's the most important part of gene linkage as far as inheritance patterns go. Right? They do not work. The Punnett squares cannot predict the inheritance patterns for these genes. Okay? The only way to separate linked genes is through crossover. Okay? So recombinant chromosomes can occasionally have linked genes separated. Non-nuclear is kind of cute, right? That's when the gene that we're looking at is on a piece of DNA that's not found within the nucleus. Okay? So the easiest example of this is mitochondrial DNA. Right? All of our mitochondrial DNA is inherited maternally. So I have exactly the same mitochondrial DNA as my mother, and she has it as her mother, and her mother, and her mother, right? Because that mitochondria is passed from the cytoplasm of the egg into the zygote. Okay? My father had no say on that whatsoever. No contribution to that DNA. Okay? So typically, our non-nuclear DNA is a single parent inheritance, and almost always it's from the mother. There are occasions, there are examples of the male, the sperm, bringing some piece of genetic information, but it's rare. Okay, so most often this is a maternal type of inheritance. Epigenetic is when I change my phenotypic expression without changing my genotypic setup. Right, so I change my phenotype without changing my genotype. And this is going to be in response to some environmental factor. And so a really easy example of this is tanning. Right? The UV light gets into our cells, it triggers the expression of more melanin. It turns on more of our melanin genes. So my phenotype changes. I get a different skin tone, but I have the same genotype. Right? So that's a really easy example. Now that you understand the mechanisms of regulation of gene expression, right, that should be really clear to you at this point in time how that happens. Right? Different transcription factors get involved, transcription occurs, the genes get turned on. Polygenic traits are simply when I have multiple genes that are coding for the same trait. Right? So there's more than one gene going at this one trait. Hair color, for instance. Right? Multiple different genes are coding for the same pigment, and they kind of add up. Right? So additive, summative polygenic traits are like the most typical one that you would think of. Right? So just think of polygenic traits as summative for the most part. And so depending on how many copies of this protein I make from all these different genes, I'm going to get a different hair color. The more protein I make, the darker my hair. The less protein I make, the lighter my hair. Right? Polygenic. And then there's epistatic, which is a type of polygenic trait. Right? Just a very specific type. So epistatic traits have two genes at play. There's the main gene that codes for the trait that we're looking at. I've used hair color.